let me just start by introducing myself, because there's probably folks who don't know why I'm here speaking today, but my name is Ben Stireman. I'm the Sovereign Lands Program Administrator, and you have previously worked with Laura Vernon, who reported to me as part of the Sovereign Lands Program. So I guess just some housekeeping. When Laura sent out her email, I actually had a lot of people congratulating me on my new position, and as a matter of fact, I'm actually just absorbing Laura's work as of now, but we do intend to fill the position. But it may take us just a moment to find the right individual. So wanted to let you know where we're at on that. But Laura is no longer with the division. She's with Water Resources, so a lot of you will continue working with her on the integrated watershed assessment. So with that, I think we can go around the room and introduce those that are here, and then we can jump online to introduce those online. So do you want to start here with you, Wayne?
Yeah, Wayne Wurtsbaugh from Utah State University in the Watershed Sciences and Ecology program.
Liv DeFreitas, Friends of Great Salt Lake. Good morning, everybody.
Bill Johnson, University of Utah.
And Chris Klein, U.S. Fish and Wildlife Service. Good morning.
Erin Rivers, Utah State University, also Watershed Science Department.
And do you want to start back there, Marissa?
Marissa Weinberg, Forestry, Fire, and State Lands.
Keith Hambrecht, Forestry, Fire, and State Lands.
Edd Hammill, Utah State University, Watershed Sciences.
Scott Heaney, United States Geological Survey.
Perfect. And Jim, if you want to introduce yourself before we move online.
Jim Harris, Division Water Quality.
Okay, perfect. And I can't see a list of everyone online, so if you wouldn't mind just piping up when you feel comfortable, you can. Oh, there we go. Paul just showed me a list. So I'll just go down the list to make it easy. But the phone number ending in 42. Okay. Alex P.
Hi, yes. My name is Alex Phillips. I am affiliated with Duke University and Great Salt Lake Institute.
Thanks, Alex. Andrew Rupke.
Andrew Rupke, Utah Geological Survey.
Thank you. Anna Bill. Oh, can't hear you, Ann.
You're on mute.
And the Village of Education.
Okay. Ann, we still love you, Ann. All right, Blake Bingham.
Blake Bingham, Water Rights.
You and Carly, I will not try to pronounce your last name. Beetle. Carly Beetle.
That was right. It is Beagle.
All right.
I'm with Westminster Great Salt Lake Institute.
Thank you. Christine Ramsey.
Christine Ramsey, USGS.
Craig Miller.
Rick Miller, Water Resources.
And Doug Ramsey.
Doug Ramsey, Utah State University, Department of Wildland Resources.
And Emily Hammill.
Emily Hammill, National Audubon Society.
Heidi Hogan.
Heidi Hoven, Gilmore Sanctuary, Audubon Rockies.
And Jennifer Biggs. Oh, Jen, oh, there we go.
Yeah, just Forestry, Fire, and State Lands.
Thank you. John Love.
John Love, Wildlife Resources. Thanks, Ann. I'm deaf now.
All right, John Ray.
John Ray, actually Jack Ray, Utah Waterfowl Association.
Okay, thank you, Jack. Kim Coburn.
I'm with the Utah Division of Oil, Gas, and Mining.
Thank you. Lauren Head.
I am with Utah Division of Wildlife, Great Salt Lake Ecosystem Program, but actually will be transitioning as a PhD student at Utah State University coming in January.
Congratulations.
Thank you.
All right, Melissa Cobo.
Utah State University, I was the master student that worked on this.
All right, thank you. Phil Brown.
Yeah, Phil Brown with the Great Salt Lake Brine Shrimp Cooperative.
Okay, thank you. Rob Hartman.
Rob Hardman with US Magnesium.
Thank you. Ryan Doherty. Dave Samdana.
Yeah, Som Datta, Utah State University, Mechanical Engineering.
And Soren Brothers.
Yep, Soren Brothers, ex-Utah State University, now at the Royal Ontario Museum.
Thank you. And then Susan Zarakrisi.
Morning, Susan Zarakrisi, Utah State Parks.
Thank you. And then anybody I may have missed that joined as we were going through that, please feel free to introduce yourselves. Okay. All right. Well, again, thanks for being here today, especially those in person that made the drive down. I know those the roads were pretty hairy, so thanks for being here. Again, Laura Vernon is no longer with our division, so we will be running the meeting here with our current staff until we find someone permanent. So I apologize for any hiccups. You'll have to forgive me if I don't run it the same way Laura did. But if there's anything that we can do to improve or anything in the future, please let us know. And we'll hopefully not be out in the Core Center for any more winter meetings. We'll try to have them in the main building. But we have a few updates to give. So Laura told me that she often gives the Great Salt Lake Advisory Council updates, and that's on the agenda today. So I thought the best we could do on that, from my perspective, is, I guess, we last met on November 2nd, and in that meeting, we did have presentations from USGS on the updated equation of state, Division of Water Rights on and Great Salt Lake, and then the Cache Water District on funding of a Bear River economic study. So there's quite a bit of discussion on the Bear River economic study, as many of you might have remembered. The Advisory Council did fund some projects for 2023. They funded the bathymetric LiDAR test plot, the Great Salt Lake Shorebird Survey, and then the Bear River economic study. And that one had a few different strings attached. We wanted to make sure that someone from the Advisory Council was involved. And so I think Laura at the time was volunteered to be involved. And so at this point, it'll be whomever serves. Yeah, me for now. I'll work that into the schedule for sure. And then we do have the Advisory Council meeting dates for 2023. So I'll just list those off so you're aware. But we'll be meeting on January 11th, March 8th, May 10th, July 12th, September 13th, and November 8th.
Wait a second, July 12th.
That's quick. Excuse me, I'm going to go through those one more time, a little more slowly.
September what?
January 11th,
okay.
March 8th, May 10th, July 12th, September 13th, and November 8th.
Is somebody going to send out invites or anything like that?
You know, I was actually this isn't on my list of updates I wanted to give, but one of the things that I'd like to immediately change that we haven't been doing is send an actual calendar invite for these. So you get them on your calendar and they're there. We have some tools that Marissa is working on that will make it a little easier. However, I think the only shortfall on that is when you get the calendar invite, you won't be able to respond directly to that email because it's going to come from like a listserv. So you'll have to be aware that you can respond to that, but when we have correspondence among us, it'll come from one of us.
Okay.
But on the calendar invites, you'll have to refrain from responding to those or it'll go nowhere. So, yeah, but anyway, hopefully that helps. I know for me, it'll help get it on my calendar and we will have to just remember that.
We
So
just set that up for this meeting too.
that's specifically for this meeting and then also for the Advisory Council. Absolutely. We'll try to do it for any of the Great Salt Lake meetings that we host or are in charge of running. So we'll get that done prior to the next meeting. Let's see. So the next agenda item is the Salinity Advisory Committee updates. Last time we met, one of the main topics that we discussed was the berm. We're currently looking at raising the berm again. And the reason being so is that if we are able to cut off flows completely from north to south and south to north.
This is the berm that's in
the causeway there. So the adjustable berm that separates the north and the south arm. So we're looking at raising that completely to basically act more as a dam. And the reason being is that all of the fresh water, as you know, comes into the south arm. And so we're hoping that by doing so, we will be able to retain some of this water in the south arm that would possibly get exported to the north arm, which will help reduce salinity and then bring the levels up in the south arm. We've done some modeling that supports that and shows that it will be effective. We're kind of fine tuning some of those things, and as you know, there's a lot of unknowns on salt balance and other aspects that we're working through. So we're making the best decisions we can with the information we have, and then we're going to continue to learn. We're going to invest in some additional monitoring on salt loading into the lake and other monitoring near the berm to help better tell that story. But those are some things that we discussed at the Salinity Advisory Committee. As a matter of timeline, we're hoping that we can actually have something done well before spring runoff. So I've been asked to have it done by the end of the year, which is pretty ambitious and unlikely. But the Corps has agreed that they believe that we can do this under maintenance of a nationwide 3 permit. And so there's about a 30-day turnaround for that. We'll be meeting with the Union Pacific to discuss any implications they might have on their causeway, because I know that might be a concern. And we also have to get a permit from them to actually bring in materials and use equipment. So there's a lot of moving pieces, but it's a priority for me to have that done before spring runoff. So we'll be really getting to work on that. So,
I mean, you're looking at drying up the north arm, and how does Compass Minerals play in?
Yeah. We've had some discussions with Compass. They're the one, North Arm industry is going to be most heavily impacted. The models, that's something that we need to discuss with Joe and others on the North Arm about what that model shows, what it would look like under different scenarios. But I was actually quite pleased with the levels in the North Arm from this action. It doesn't have as much of a drastic impact because evaporation is less on the North Arm because of the high salinity. And really the only withdrawals are going to be, from extraction. So those are things that hopefully we can share in the next week or two. I hope that the models, I actually met with Water Resources, who runs GSL for us a lot of the times on these, and that was just yesterday. So they're going to give me some of the data today, hopefully, and we'll be able to better represent those numbers and show exactly under different scenarios, flow scenarios, what that might look like.
What will be the elevation of the berm when you?
Well, those are things that we're modeling right now. So what we'd asked was if there's a good target that we should shoot at. I had suggested 4192 because I think that supports perhaps getting to that microbial light ring that we all know exists, and perhaps salvaging some of the desiccated microbial lights if that's even possible. But that's an elevation that I think would be great. But the other thing that we need to account for is what the salinity would look like if we had all the fresh water taking it up to 4192. It might actually get to a point where we may need to be concerned about the salinity becoming too low if we're keeping all the fresh water at that side. So those are things we'll have to monitor, and that's another reason we want to up our monitoring, is to make sure that if things aren't going as planned, if salinity is dropping too quickly, that we can react very fast and go in and modify that berm again. And the last time we modified it, we did spend some of the money to make sure there was good access getting down to the berm so it can be easily modified in the future. And I think the gold standard, of course, would be like something we can change on a daily basis to when we need to. Yeah, but it's pretty complex when you're dealing with such saline water and remote locations and things. So we're working with rocks right now.
Thank you.
Yeah. Any other questions on that?
Yeah, so the 4192 would be roughly a 3-foot head difference between south and north. And we've had that before during the closure during 2014.
That's correct.
17. So Union Pacific, we know that it can withstand that over at least a certain amount of time. Is their main concern the stability over longer time?
I haven't had the discussion with Union Pacific, or excuse me, the specific discussion with Union Pacific, specific to this exact issue, but there is. So I think that was part of their concern previously, was that the head difference was causing some issues. And so that's something we need to be sensitive to moving forward. And so that's the question, is we need to ask them exactly what the problems were and if there's ways we can mitigate. Christine Ramsey, who's on here, had made a great suggestion last time about perhaps when the elevation gets to such a level that it would support this, we could perhaps export salt through the berm by lowering the berm at times. And that would be a benefit because, at certain times of the year, we could push water to the north arm, which would make that difference in head elevation not so great. And then it would also send some of that, sealing water to the north side. So there's different scenarios. Again, this is one of those things where we are working very rapidly with the best information we can. We're hoping to make some changes that we know will impact them, with the resolution that if we need to go in and change things again, we're very willing to do so. And we're going to work with the folks who have the science and try to get better monitoring so we can tell that story better and know when we have to react and when we can be proactive as well.
Yeah, that's it. You call it an adjustable berm, which I think is a great term for a pile of dirt.
Yeah, exactly. Yeah, it is interesting, and I think it's a great tool that we have. And I mean, to be quite honest, that we're in a scenario now where if that berm didn't exist, we didn't have that tool available to us. So I'm thankful that we're able to use some of these tools to make a difference under these circumstances that we've had with this drought. So anyway, any other questions about online or in the room about the berm? Or the advisory or Salinity committee? Okay, perfect. I will move on to to item number C under the updates. It says here Great Salt Lake Tech team member updates. So I think if we just want to go around the room to the tech team, if anybody has any updates they would like to provide. I don't know if that's allowed. Laura has always done it, but if anybody has any updates they feel are relevant, now would be a great time to provide that. Okay, I'll take silence as that I don't need to do that again. So there's a sub-bullet under there about hot topic requests for fiscal year 2024. This is one thing that I would really appreciate any help on, and that is just if you have ideas for hot topics, it would be fantastic if you could send those, even if they're not something that you have in the docket for research. At this point, if it's just an idea, we'd love to hear about it. But if you have research proposals and things that you think would make a good hot topic, now's the time to send them. I made a request to get additional funding for hot topics in my budget request this year. And so we're hoping to more than double the hot topic funding, as long as that goes through. It looks to me like it made it into the governor's budget, but it's hard to pick out line items like that. So hopefully we receive some additional funding for hot topic grants. One of the reasons that we did that is we're hoping to do some larger studies as well. Historically, we've funded several projects, but I would really like to have the opportunity to do some more in-depth projects that maybe were prohibited by cost. So more to come, but I wanted to mention that because I wanted to make sure that when you're coming up with your funding requests or hot topic grants, hot topics, that you keep in mind that there may be additional funding this year. So if you have those larger projects, it would be good to throw them out now, and then by the end of the legislative session, we'll know if we receive that extra funding, and hopefully we won't have to come back and ask for new topics. We can work with what we have and just offer those that way. When we do normally in April, I think is about the time we'd send out those RFPs.
So when's your deadline for getting these hot topics from us?
Well, I probably need to give that a little bit of thought on our timeline and see what Laura's done in the past to make it consistent with that. So if it's okay with you all, I can send out an email after the meeting with specific deadlines.
And then, so the funding is typically announced in?
I believe we rank the grants by May, so the RFP would probably be coming out. Again, these are things that I hadn't.
RFP or
the request for proposals for. I guess as we identify hot topics, you'll put a proposal in for your grant.
Okay, okay.
And
so the ranking is just for the topics, not proposals.
So right now, we're just asking for hot topic suggestions. So research topics that we would want to rank as our hot topics. The way we've ranked them in the past is we accept grant proposals based on the topics that are identified. So we're not just accepting grant proposals on any subject on the Great Salt Lake. So they're very targeted. So now's the time to come up with those topics and bring those to me. And I can provide a timeline shortly after this meeting. I'll look and see what we've done in the past and see if that seems like it made sense. And if we need to adjust anything, we will. But otherwise, we can plan on a similar timeline. Bill, do you want to?
Yeah, so we've been trying to ratchet it forward in time a little bit so people can capitalize on the early realty. They're actually out in May, and I know that the funding has this July timeframe, but still, we try to sneak in work, because you gotta do it when it's summer.
Yeah.
And so if it's possible to shift the RFP a little forward to like March, I think that would help people.
Yeah, I don't know that it'll help the funding very much because the funding has a very hard deadline or start date of July 1st.
Yeah.
But knowing and being able to plan, I can definitely see the utility in that. And the last thing we want is to have any short-sightedness. So I'll attempt to have the deadline set earlier. I'll look at what we've done in the past, and then if we need to make some adjustments, if y'all have some suggestions, I'd be open to doing this earlier, so you have more time to prepare. That'd be fun. And actually, that helps because May is when we really start to ramp up. So if we can rank grants in April, that might be better timing for us too. So, Tom.
Earlier than that. When we submit a topic, I assume you want a paragraph rather than a one-liner, right?
Yeah, paragraph would be great. So the general topic of what that would look like, and then some explanation as to why you think that it needs that. Thanks for that, Tom. Perfect. Any other questions? It looks like there's a few comments coming in on here. So. Okay, so it looks like there might be different dates in the agenda.
Those dates that you announced, were they for the advisory council?
Yes, those were the advisory council dates. Is that the confusion? Are these look like these are the tech team meetings?
Yeah.
Okay. Perfect. I will send it. So just to clarify, I'll send a follow-up email after this that clarifies the dates for the tech team, the advisory council, and then any of the significant dates for the hot topics. So there will be a follow-up. And again, I apologize. This is I wasn't necessarily expecting to be running these meetings as of like 2.5 weeks ago.
Right.
And I've had a lot to balance with other duties. So kind of death by fire here, getting and picking up where Laura left off. Thanks, Ann. And thanks. Yeah, appreciate that. So anyway, with that, you've all heard. Yes.
Just a quick question on that, just as kind of a reminder for people who aren't as familiar with them, and and I'm trying to communicate information to people, so I just want to make sure that I'm clear too. The hot topics are research oriented, or are they management oriented as well?
Research oriented. So a good place to start if you have questions about what they may have looked like in the past or topics that have already been studied would be to go to our website, and there's a list of previous grants that have been awarded, and then also like last year's RFP that had the hot topics on there, so you can get an idea of what has been done. And it's come to my attention that some of those final grants have not made it onto our website as they should have. So we'll be working to update that, but that should give you an idea of where or how that works. Any other questions about the hot topics? Okay, well, thanks for bearing with me. I will move on. It looks like we can go to our presentations, reports, and discussions. So we have Edd Hammill from USU here to talk about metal concentrations in wetland plant tissues, influences transfer to terrestrial food webs. So Ed, take it away.
Where do you want me?
Do you want to come just sit at the table, wherever you're most comfortable? Do you need to?
I like pointing at things, but I realize that's not
Yeah, you can wherever you're comfortable.
Sure.
Perfect.
We got a kicking right, just the humor. Just the what? Next slide, please.
Oh, there's lots of animations.
So can I do the?
In that case, it's next animation.
That's why it's got kicky kicky. Sweet, thank you so much. Oh, sorry,
Jake.
This is a group right here.
Hardcore. Okay, well, yeah, thanks. Hi, everyone again. I think I recognize some faces in here. It's good to see some new folks as well. My name's Edd Hammill, Utah State University, and I'm talking today about the results of a project that was actually funded by one of the Forestry and State Land grants a few years ago. We recently completed the paper and the news media and all the other stuff associated with it. And the title of it is Metal Concentrations in Different Plant Tissues Influences Transfer to Terrestrial Food Webs. Before I start, this was not just me. This was a big team of Tricia Atwood, Karen Kettering, Janice Brainey, and Maya Pendleton, all from Utah State University, 3 professors and someone that actually did the work. So the reason we got interested in this area at all about metals is because metals are persistent toxins. The difference between metals and a lot of other toxins is when metals are in, they stay in. It's very hard to then get them out again. They don't get used up, they don't get metabolized, they end up persisting in ecosystems. And while there are some metals naturally present in the soils, the Great Salt Lake does have some metal inputs from things like the Kennecott mine and also refineries. As a result, we've got higher than what you'd expect naturally. And our question was like, well, where do these metals end up? If they're in the soil and they stay in the soil, is that really necessarily that bad of a thing? Or what happens if they end up in food webs? And what are the potential mechanisms by which they can get in food webs? And one of the reasons we really looked at this is because one of the metals, mercury, gets a lot of attention because there is still the consumption advisory for birds on the Great Salt Lake because of metal contamination. So we thought it might be something worth looking at. So the first step we looked at is, well, metals originate in the soil. What influences transfer from the soil to the entire rest of the ecosystems? The first step is the plants. So we've got these wetland plants, things like Phragmites, hardstem, 3-square, and alkali bulrush. They're growing in the soil. The metals are present in the soil. They can then be absorbed into the below-ground tissues, things like roots, bulbs, and rhizomes. From there, they can be transported into the above-ground ecosystem where they hit leaves, stems, and seeds. And these plants are classed sometimes in 2 different ways. So you can call them excluders, and for excluders, they take up metals, but they limit them into these below-ground tissues. So as a result, although they're technically now part of the biota, they're remaining below ground, and they're not really going anywhere. On the other side, you've got accumulators. And what accumulators do is they transport these metals from the below-ground tissues up into the above-ground tissues. The issue with that is then you have a sort of double-edged sword. In the process of that, there is the potential for those metals to now be available to the terrestrial ecosystem because herbivores can eat them and things like that. But on the flip side, this is actually used as a method of bioremediation in some parts of the world, because if the plants are sucking the metals out of the soil and putting them into the above-ground tissue, you can then harvest that above-ground tissue, take it somewhere else, and as a result, you're extracting the metals that were previously in the soil. So although it can be a bad thing, because it can be moving these things into the terrestrial ecosystems, it can also be a good thing because it means you can extract them yourself and take them out. The only issue is that different species will behave differently for the same metal, and one species may act differently for different metals. So there's no like smoking gun magical plant that does everything for you. You have to kind of get the metal approach. So what then happens afterwards? Well, the next step is trophic interactions. So when the metals are in the plants, you have the situation where insects, grasshoppers, things like that, they consume the plant tissues. As they're consuming these tissues, they're ingesting any toxins that are there. You can then get what's called bioaccumulation. Bioaccumulation occurs within a single individual when, if the toxin or metal that is present, if that binds to the tissues in that insect and it's not excreted, it means that that insect is continuously carrying all the metals it's ever encountered in its entire life in its body forever. That's not bioaccumulation, accumulation within an individual. Worse than that, you have biomagnification, where what happens if that insect is then eaten by something else, for example, the wee spider at the top, that spider is then getting a dose of all the metals that insect ever encountered in its entire life. So you can have the situation where as you go up the food chain, the concentrations are actually magnified because every trophic level up is getting all of the metals the trophic level below ever encountered in its whole life. This hasn't done. Okay, so we found from a previous project that plants in the Great Salt Lake grow in these large monotypic stands. So what this means is you're not getting a mixed community. You just get these big areas, massive areas of Phragmites, big areas of cattail, things like that. Previous project, we actually mapped these out using NDVI and ground truthed it. And so you can see the color scheme. So as a result, we have white as the dominant landscape, but we've also got dark green as the Phragmites. The orange is things like pickleweed. We've got 3-square. what this means is we can not only quantify what the metal transfers are within a plant, but we can actually start to quantify it on the landscape for whole areas. So it's something to think about not only can you use different plants to do different things, you can identify in the landscape where these processes might take place. We generate, with these maps, we do exist. It's from a previous project funded by Forestry and State Lands. This is the migrated bird refuge, same sort of map for the TNC. So we can not only use these maps to identify where to work, we can use the maps to identify where these processes can take place. So we then started to ask ourselves two big questions. So the first one is, do different plants exhibit varying concentrations of metals across their tissues? And do metal concentrations increase in terrestrial species? This first part is asking, are these plants acting as accumulators or excluders? The second bit is asking, are we seeing biomagnification and bioaccumulation existing within the ecosystem? So we picked out a bunch of sites. This is taken directly from the paper. We looked at 4 particular species: hardstem, alkali, Phragmites, and 3-square. These are our sites dotted between the migratory bird refuge and then the TNC. So we went out to every single one of these sites and we collected a ton of samples. So for all of the different 4 plants, we collected, I've done these a little bit by different colors and attempt to group them into trophic levels. So these are our predators: spiders, dragonflies, damselflies. We also collected just flying insects, a random sample of what was just flying around, so that's mosquitoes and everything else. We looked at the grasshoppers, slugs, and snails and caterpillars. These are the herbivores that were collected directly off individual plants. Then in the plants themselves, we took samples of seeds, leaves, stems. Those are the above ground tissues. Then we did the rhizomes, roots, and bulbs, below ground tissues, and finally the soil. So an absolute ton of samples. Oh, are you right? Some of them, so going out for the grasshoppers and the odonates, the damselflies and dragonflies, I literally went out with a net and ran around grabbing hold of damselflies and grasshoppers, which was kind of fun. And these are what we call window pane trapped flying insects. So you have 2 bits of glass set like that, or 2 bits of perspex. The bugs, as they're flying along, smack into those, then fall down into the funnel and get caught in a little thing down there. As a result, you get a sample of what's flying around in the actual ecosystem. The reason we looked at it this way is because these plants grow in these large monotypic stands. So for things like grasshoppers, slugs, and snails that have really small home ranges, and spiders that have a home range sometimes with an individual seed head, you can get the idea that the insects that we're catching there have been feeding in this ecosystem, and that's where they're from. You're not getting a lot of immigration. The window pane traps, it was all right to set up in the Bear River Migrated Bird Refuge because you can drive to most places. When you have to carry them for kilometers in the TNC, that was slightly less fun. They do weigh quite a bit. But we collected all these samples and then they were prepared, and we're looking for concentrations of arsenic, lead, copper, cadmium, mercury, and selenium. So when we get through to the results, there's going to be an awful lot of figures and graphs. And what I'm going to do is show them all and then we'll work through and compare down. So just a reminder of all our different sites. We had 4 different metals, 6 different metals, 4 different plants, and about 9 different tissues.
And 2 sites.
And 2 sites, yeah. So a lot of data. First thing we found out when we looked at soils, so on the x-axis here, we've got the 4 different plant types. The y-axis is soil concentrations of all our different metals. The thing that really jumps out straight away is that Phragmites seems to be found in soils with very high mercury concentrations. Now, we don't know if that's because Phragmites is better at outcompeting in areas with high level of mercury, or if it's because these other plants are actually extracting the mercury out of the soil. We can't say that. You can't infer causation from the correlation, but generally found enormously high concentrations of mercury in Phragmites soils. Other big ones, 3-square and Phragmites seem to be able to cope with high levels of arsenic as well compared to the other 2. Apart from that, a lot of the other metals are actually in relatively low concentrations in the soil themselves. But this is what we're starting off with in the soil. This is just what's there, not yet biological.
And just clarifying, this is wetland soils, not playa soil.
No, we're literally, so when we're taking these samples, we're going to the plot. We have our plot is a GPS point, and we literally dig up a great hunk of the soil from there. So now we'll go through them in bits. these are arsenic concentrations first of all. We have here the brown is the below ground, the green is the above ground tissues. This is in the terrestrial food web, with the sort of redder colours being our predators as we work through. So if you look at arsenic concentrations, first thing that jumps out straight away, there's much higher concentrations of arsenic in all of the below ground than there is in the above ground. So it looks as all the plants for arsenic are acting as excluders. They're not transporting arsenic into the other parts of terrestrial food web. Also, we didn't find any arsenic at all in the insects or the invertebrates, which in a way is kind of good. Then we have lead. Big thing about lead, very low concentrations in the below ground for most of the plants. Some instances of very high concentrations in the above ground, especially Phragmites in the seeds, has massive concentrations of lead. So that appears as if the lead that's present in the soil is being transported up through the plant, making it into the seeds, and that's where it's sitting. Okay. In other ones, we saw some evidence of biomagnification. So if you look at these are our predators, and if you look at the concentrations of lead in the predators compared to any of the plant tissues, you can see you've got in some cases 5 to 10 times more lead in the insects than in the plant tissues they're associated with. That gives that indication of biomagnification.
A quick question.
Sure.
So, in terms of very high, those statements, I'm assuming you're saying relative to other concentrations, not relative to what's found in other settings.
No, so this is relative to what, remember, this is all what's taking place inside the actual Great Salt Lake ecosystem. So it's all relative. It's not like this is high compared to if you're choking down diesel fumes.
So but you're basically like in Phragmites, you're seeing something on the order of 15 to 20
times what's in the background,
What is it, micrograms per gram?
Yeah,
So
micrograms.
that's parts per billion?
Yeah.
ppm, isn't it?
Okay.
Yeah.
Is it ppm or ppb?
ppb, I think.
A micro, you got millionths of a gram in a gram. That's
ppb,
right.
Yeah.
No,
ppb.
ppb. I'm thinking
ppm.
It should be ppms. Yeah, ppm. Sorry.
Okay.
We got shouted at for putting it in ppm in the original paper and got told to put it back to
Just trying to compare that to some of the X levels.
And also, what would be interesting to see is the end values for those different standard deviations that you're showing.
Oh yeah.
Just to understand, whether some of those particular.
Oh yeah. So how many?
Are,
so in every, by and large, we try to get 9 samples and everything. So that's 9 sites. Okay.
Yeah.
In some cases, you couldn't always find them.
Right.
I mean, there's some things that just don't. Some in, like hardstone doesn't really have leaves. Is one of the things. There's these various issues. But what we do, we literally collect as much tissue as we could, and they go for a minimum of 9. Some of these get really wide. I mean, you can see the Phragmites seeds do get really wide, so that's possibly the situation where there was one site where there's just enormous amounts. But generally, I mean, even the tightness of that.
So this is averaged over all of the sites?
Sites, yeah.
Oh, really? Okay.
And Bill asked online if this was done by weight. She commented that Phragmites seeds are much larger than alkali seeds.
No, this is a concentration. Yeah.
In the seeded soil.
Dry weight?
Yeah.
Right.
Yeah. So there's you had to, we literally, for every, the way that samples are processed, you take the tissue samples, get the dry weight, you have to hit quite a high dry weight for the sections to run, and then you smash it to a powder using a coffee grinder, basically. So we went through about 40 coffee grinders. It was pretty interesting. Copper, we see some cases of the same sort of situation, low concentrations in the plants, very high in some of the predators. So again, some evidence, but again, not super clear. For cadmium, we found some of the biggest incidents of biomagnification in the whole project. So if you look at, this is the plant tissues in alkali for cadmium. This is then the spiders that were found living on those plants. So in some cases, that's 10 times higher than what's in the plant itself. So that's a spider that's living often like in the seeds. You literally, as you're going around collecting the individual, collecting up a whole bunch of individuals, grinding them down, And then as a result, you put the cadmium, the amount that we're finding inside the individual is really nasty.
Because spiders are basically predatory, so you
Yeah.
think they're getting it from.
Yeah, so what's happening is that situation, the things that are eating the leaves on that plant, the spiders are then consuming, so they're getting that biomagnification.
Unless they're insects that are getting soil exposure.
Somehow, yeah.
From the soil, okay.
One of the really interesting ones was mercury. We didn't find mercury in any of the terrestrial invertebrates, none at all. We did find massive amounts, well, relatively large amounts in the above ground tissues of all the plants. So we didn't find any it was below the detection level for all of the below ground tissues, but it was way above the detection level, actually relatively high for some of the above ground tissues, which suggests that for all these species, from mercury, although the concentrations are quite low, it didn't look like they were being transported into the terrestrial food web. They are accumulators that are then bringing them up into their above ground tissues.
So the scales changed there, right?
Yeah, the scale's changing.
Well, they're tenths of ppms.
Yeah, so they're quite low.
Okay.
Okay, so Ed, just quick on that. You know, multiple groups have measured the spiders for mercury and seen astronomical concentrations.
Yeah.
And so I gotta wonder what's going on here. Talk more.
Yeah, I mean, my thing with it is the concentrations of mercury. Its general it gets an awful lot of attention because at very low concentrations, it's relatively nasty compared to some of the other toxins.
Yeah.
We just didn't find it, and I don't know if that's because potentially we didn't get enough tissue.
Sure, and that's what I'm wondering is
Yeah.
this ICDMS analysis of these tissues?
Yeah, this was, we chucked it over to the place at University of Hawaii, Hilo, and they told us how much we needed for every single metal. And I suspect, to be honest, we just didn't give them enough. It's a big thing.
So
yeah, we should talk
what,
more.
Yeah, what were the detection limits on?
Low. They were below basically 0.01 or 0.1.
Okay.
But, you know, ppm is massive concentration
Yeah.
for mercury.
It's huge.
Right? We usually measure nanograms.
Yeah. So my thing would be, if we were to do it again, sometimes trying to actually get enough tissue, some of these things, like it's, for the plants, it's not too easy. For them, when you're trying to get spiders, like getting grams and grams and grams of spiders, and the...
But you don't have to, and we can talk more about
That'd be
the
great.
analyses that can be applied here.
Awesome, yeah, because we've still got these.
I'd like to talk more about
Yeah, sweet. Because that was a weird one. And then last but not least was selenium, which again, we had that same situation of lots of it in the below ground, hardly any of it in the above ground. So it looks like, again, that's being kept. So, just go through again the main parts, I thought the big takeaways. So, although Phragmites was found in high mercury soils, as we talked about, we didn't see any transfer to the above ground terrestrial food web. Whether or not that's because we're way below the detection limit, the fact that there was so much in the soil and then we're not seeing it anywhere else was kind of surprising. So there might be something else going on in there.
So just in terms of soil concentrations, were you seeing big differences in mercury concentrations in soil between your sites?
Yeah, I mean, well, these again are the averages of all the different 9 sites. The nice thing about soil is that we absolutely got enough material every single site. The soil uses a slightly different analytical technique, but what surprised me was if you look at the actual concentration, remember this is the soil concentration across all the metals. Some of the mercury concentrations we found in the soil were the highest concentration of metal we found anywhere at all. Whereas when you look at the amount, I mean, this scale is different on the y-axis. So although you've got some of the highest concentrations of any metal is inside that mercury, it doesn't seem to be going anywhere else, which was kind of bizarre. So.
Yeah, I mean, because you do measure high concentrations of mercury in soils.
Yeah.
And the question is whether, how much of that is bioavailable?
Yeah.
Methyl mercury.
Yeah, methyl mercury, yeah. We went with straight mercury, and I think, I don't know, I just asked that, I think methyl mercury would be the way to go again.
And that could actually be a factor in Phragmites, is that the soil chemistry is such that it's a more reducing environment, either because of the Phragmites or that's what grow in.
Yeah.
That would tend to give you higher methyl mercury.
Yeah, I mean, there was a massive discussion that went on when we first did, because we got to choose whether we were doing methyl mercury or mercury. We ended up going with, I can't remember exactly why we landed up with the regular mercury, but the methyl mercury, I think, I mean, the toxicity of methyl mercury is much higher as well.
Direct storage.
Yeah. But so that was a weird one. So this was the bit that was most like confusing and concerning at the same time. The fact that the highest concentration of any metal we found in the soil was mercury. Then it was not.
But you only found it in the phragmites.
Only really found it. I mean, there's bits of it, but not what you call a lot.
Freddie had a question at the bottom.
Oh, sorry. How do you separate other sources of metals during the aquatic phase of life cycles in the ozonates? Thanks. Yes, so the big thing is, the question I'm asking is about the odonates here, they start off in the water. We weren't able to separate that out. One thing I'd love to be able to do is actually get the dip net out and go, because the nice thing about odonate larvae is they're quite large, so you wouldn't need to catch that many of them in order to take a sample of that. Having a look if there's a difference at different life stages would be a big point, or whether or not getting it all in there.
Well, and kind of the difference between odonates and spiders would also kind of imply that difference.
Yeah. But one of the big things is the spiders sometimes are so much higher than the odonates.
But the spiders, as you mentioned, are very low territory. The dragonflies particularly migrate over great distances.
Yeah, they can move around, yeah. But when that's why I think that the larvae are a good way to go. Because then the larvae are just like, they're not flying anywhere. So that's a good way to go. One of the stories that was quite nice was all plant species retain arsenic and selenium in below ground tissues. So there's no, from what we could see, the concerns related to, is it getting transported into the above ground through plants? Not necessarily. There is this weird hard stem spider. This was one spider that was just absolutely chock full of selenium. So I don't know what it was.
So that's an N of 1.
Yeah, it was like, yeah.
It's a necessary micronutrient in humans, right?
Yeah, no, but it was just.
Maybe it's a bigger standard for spiders.
Yeah, so.
Can I?
Yeah, sure.
So are these excluders, do they truly exclude these metals? So like, are there other ways that these metals can be transferred into the food web? Like are things eating below ground tissue that then goes?
There's a little bit of consumption of below ground, and there is the breakdown when the thing dies. It goes, but then, to be honest, from what I understand, and Janice was the expert on this, and I wish she was here because she'd correct me when I'm wrong. But from what I understood, you've got the below ground tissues, and then the metals are ended up in there. Then when the plant dies, those below ground tissues often rot below ground, and so it just gets transferred back into the soil. Erin's nodding her head. Is that? Yeah. Okay. Feel better about myself.
But then there could be some questions of like how available those metals are, because if they're bound up to the organic matter and then
Yeah.
you know they're there, but they're not as available for uptake by other plants or organisms.
Yeah, I guess I would see. Yeah, C3 goal.
And that would be metal specific too. So some metals are better than that.
And then the last bit was, we did find some evidence for biomagnification, but no clear patterns. So these are the ones I pointed out. Again, in all of these instances with the predators, we're getting metal concentrations that are much higher than are seen in an awful lot of the other parts of the plant. So you have got this evidence for it in cadmium, lead, and copper. Lead was the most extreme. It was found in basically every ecosystem. There was still this weird amount of lead in Phragmites seeds. But for lead, for every single plant type, you're finding evidence of biomagnification up into the predatory guild. Finding in half of it for copper, a couple of times for cadmium. But what was interesting was there's no just clear pattern. So lots of these, there was no one plant that biomagnified ecosystem that every metal ended up biomagnifying. Lead was seen the most often, these two and the other two, but it's not. You had to do it on a sort of case-by-case basis, which was the hard thing. So as I said, yes, some potential for bioremediation. So because there were some instances where you could use plants to transport these metals into the above-ground tissues for harvesting. But if you're going to try and use that, I wouldn't necessarily extol it as a management technique because it's very metal-by-metal, plant-by-plant basis. Doesn't feel massively viable. There was some evidence for some trophic transfer, so there has the potential to be a concern in some cases, but some of it was still not actually concrete. So then my next question was, and this is nice that the tech team's coming up, where next? So we identified one potential route where you could be moving metals from the soil and the lake and generally into the terrestrial ecosystems, but then we started to think about there is another potential route which we totally overlooked, and that is some of the fish. So there are fish in the Bear River Migratory Bird Refuge, there's fish in the wetlands, there is invasive carp, catfish, and a few other things. Especially with the refuge, the way that some aspects are allowed to go dry and some areas are kept wet, that means that sometimes when we saw it when we were out there, you'll have large amounts of dead carp just lying around. So these carp, after drying, they turn into this. So then what happens to them is our question. So there is anecdotal evidence from other places that there are a whole bunch of scavengers that opportunistically will harvest dead carp. So coyotes will take them, raptors will take them quite happily. Our concern is then, all right, if you look at the total amount of metals present in the ecosystem, this is a tiny proportion of what's getting moved around, but it's getting moved around in a very specific way where these metals are not universally distributed around the ecosystem, they're concentrated in these massive packets of protein. So you might have a situation where even if the overall concentration is not very long, if this carp has been swimming around and it's been consuming and absorbing metals its whole life, it then dies. It means this coyote or this raptor is then getting a massive dose of these in one go. So what we'd like to do, I think, for a nice idea, quantify the concentrations in these species, quantify the concentrations in the dead ones as well, see if there's any sort of decay, set out a bunch of camera traps after somewhere's been decayed, have a look at who's eating them, see what's going on. But just as a very vague idea. We identified one route into the terrestrial ecosystem. This might be another that totally circumnavigates the rest of the ecosystem and just goes straight into the top predators. Okay. So, thanks for your time. And any questions?
Did you ever consider buying a steak from any of the grazers that graze on Phragmites?
No, you see, let's have a look. You know, you see things like the amount of seeds that are present, the amount of lead that's present in seeds. Sure, it's like, no, I'm all right. No, even not red meat in January anyway.
In terms of the fish transfer, I mean, there's a lot of birds that eat the live fish too.
Yeah.
Pelicans, obviously, great blue herons, and so forth.
Yeah, my reason for aiming at these, there's a couple of reasons as well for going for the stick. So I'm not totally au fait on pelicans, but I don't think they eat massive, gigantic carp, do they? Really?
Everything they can fit in their mouth.
Because my thing with this is, if you think about how some of these toxins work, especially with that bioaccumulation, if it's a great big fish that's that big, it's now containing all of the toxins it's ever encountered its entire life. So the concentration of some of these toxins per gram, is much bigger, that big. If something is eating a kilogram of tiny little fish, it's getting a much lower dose than if it eats a kilogram of one gigantic fish because of that accumulation through time. Okay. Anne, did you raise your hand? Don't. Definitely.
Yeah, thanks. Is this better?
Yeah, it's great.
Okay, sorry. Just so you know, Kennecott's done a lot of studies on bugs and eating and mostly aquatic stuff. And I think they're all public because they were part of, Chris, help me out. What were those studies?
What's that?
The studies that Kennecott did, CERCLA or...
Yeah, definitely for the Superfund, the CERCLA stuff in the North Zone wetland areas.
Right. Right. Anyway, that might actually shed some light on some of the transfers and trophic type of thing because I agree that I mean I think it's great if you want to continue on with the larger animals and that trophic uptake, but I think there's a lot of information out there that is not published per se, but it's definitely been reviewed by EPA and other people.
The other thing about some of the larger animals, and this was stuff that's been done actually by Tricia Atwood, who's one of the collaborators at USU. She's done a study on looking at seed dispersal like coyotes, and the fact that although if you look at the total amount of stuff they're contributing, some of that's going to be quite low. They're doing it in a very different way. It's massive concentrated ions rather than so things like, say, if you're concerned about these metals entering the ecosystem, this individual coyote might eat a whole bunch of get a massive dose of something from these carp. when it then goes and excretes somewhere, and often these things use latrines, and that's a very small area potentially about very high concentration. And it's different than something being uniformly spread over the landscape.
So a couple of things. One is that the data's a little old now, but Fish and Wildlife Service in the late 90s, early 2000s did a pretty comprehensive, contaminant screen of all the wetlands around the Great Salt Lake. And so that report has data on metals levels in carp.
Oh, wow. Okay. Yeah.
The other thing is that in terms of, that picture there, I would pay attention to like how desiccated or how decayed these animals will eat those, because those will affect the concentration that they're getting. And then the third thing is that I think you might want to take the data that you've got now and like compare it to some of the effects levels. For example, how much mercury is an adverse effect in spiders, or how much is enough to provide a dose to another receptor. And if you want to get in touch with me, I can help you.
Yeah, because I think that desiccation aspect is a huge.
Yeah, because I don't know that like an eagle will, they tend to eat something that's more like they'll concentrate on the as the carp hit the salt water and then they die on the salt water. The eagles tend to nick them now. They're not going after ones that have been sitting out there for 3 weeks.
Because I think, ideally, the way that you would do it is literally do it when they're alive, get it immediately after they're dead, and then stick it to one side and let them decay both. I'm sure they love this in the
the analytical chemists love getting 5 pound,
highly decomposed.
We used to send them like literally coolers full of carp, and they would have to, just the protocols were, you have to composite 10 carp, so you got 10 3-pound carps, suddenly you got a 30-pound composite. They want this all. So.
Yeah, and then measure it through time.
Big coffee grinder.
I think in order to get on to the other 2 presentations, we better move on to the next one so we have some time here. So the next presentation is Erin Rivers and Doug Ramsey, which is titled A Multi-Tiered Assessment of Primary Production.
Yeah. Yeah.
We were using that to
Chris, what are those levels of sizing levels?
I think I couldn't see enough there, and I don't have a little committed to
Looking against the drinking water.
Right. Well,
So I'm actually only going to be speaking for a few minutes, and then I'm going to hand it over to the folks that actually did the work and about. So our project is titled A Multi-Tiered Assessment of Primary Production in the Great Salt Lake. And this was an interesting team situation. So the original PI on this was Dr. Brothers, and we unfortunately lost him to the Great North. So he's currently at Royal Ontario Museum, and he's with us virtually today. So he's going to be presenting a lot of the results for us, as well as Dr. Ramsey, who did a lot of our GIS and remote sensing work. But I think we have the whole team here today. Melissa Cobo was our master student that did some really grueling field work to make this project possible. And then Dr. Wurtsbaugh, of course, is an advisor on this project. I'm Erin Rivers. I am also an assistant professor at Utah State. My field of work is generally biogeochemistry and water quality. And so Dr. Brothers is our limnologist and expert on phytoplankton and primary production. So he's going to give us a great talk today. But briefly, I apologize for the typo that I'm just now seeing. I'm going to let Soren will describe in depth more of the actual methodology. But in general, this project was an attempt to provide kind of the first comprehensive quantification of primary production in Great Salt Lake. And so previous estimates are pretty dated by now. The 1970s, we had some estimates, but the lake itself has so many challenges that make this a really difficult endeavor. And so this project had 3 different methodologies by which we were quantifying primary production and ecosystem production. The first of which was using SOND data to measure the changes in dissolved oxygen in the lake and putting that into a model to determine estimates of primary production. The second method was using various different compartments of the lake to look at primary production from benthic communities, from phytoplankton pelagic communities, and macrophytic communities. And then the last, which will be what Dr. Ramsey talks about, is using satellite products to remotely sense chlorophyll changes throughout the year. And with that, I'm going to turn it over to Dr. Brothers, and he will show us a little bit more in depth on methods and then some of our awesome results.
Thanks, Erin. I'm going to try sharing my screen. Let's see. Okay, is this working? Can everyone see my presentation now? And hear me all right? Okay, I don't see any of you, but I'll assume that everything is good in Salt Lake City right now. It's great to see some familiar faces there.
Amazing here.
Miss you all. So anyways, yeah, thank you, Erin, for the introduction. And I'm gonna go through, it's not too much, I mean, some of this is going to be a little bit of a recap of what Erin just mentioned, but I figured I'd jump right into what we wanted to consider in terms of the primary producers in Great Salt Lake. So the work that had been done, Erin mentioned briefly, there's a Stevens and Gillespie paper in 1976, and they were just looking at phytoplankton. So, and historically, that's what people consider for lake primary production. So it's the algae that's floating in the water column itself. And that's so that was something that we considered looking at primary production, but we were also interested in seeing the benthic algae, so what we call periphyton, which is attached to the sediments or attached to the rocks, to add that component to what's been recorded previously in the lake. We might consider periphyton also to include the microbialites that are in there. They're cyanobacterial, and most of you probably know about them. But I considered them as well, sort of as part of that benthic algae group. And then we thought about submerged macrophytes, which are in the shallow bays. These kind of, I mean, in this picture, it's in Farmington Bay, but there are a lot more in Bear River Bay, just to kind of get a sense of what these different productive groups actually contributed to the whole lake balance. One thing that's sometimes contentious in people doing primary production work is whether to count emergent macrophytes or floating leaf macrophytes with primary producers. We did not include them here as part of the production of the lake. You know, basically because the methodology we use also really looks at aquatic oxygen dynamics, and these are exchanging their gases with the atmosphere. So I consider them to be kind of a terrestrial plant that's delivering organic carbon from the atmosphere to the water. But that's just something to keep in mind when we show the results. Erin mentioned this briefly, but I'll kind of go over it again too. In terms of how, so the reason we wanted to actually have it as a multi-tiered assessment of primary production is because primary production is actually really tough often to get precise measurements of, even though it's a simple concept of how much organic matter is growing, is accumulating and being produced in the system. Often, historically, people look at, say, chlorophyll as an analog of primary production, but that's really a biomass. So you think about, I had one colleague who would say that, you know, estimating primary production from chlorophyll biomass is kind of like estimating the speed of cars based on a snapshot of a picture of a road. You don't know how quickly they're going or how many cars are going through with the rate, but production is a rate of change. And it has relationships to biomass, but it's not the same thing. So one of the main approaches that we did was to look at the diel oxygen curves. And just for those who aren't familiar with it, if you put an oxygen sensor in the water, in any lake or river, typically you'll see something like this. So each of these peaks is growing up in the daytime, and then at nighttime you have the oxygen going down. And that nighttime, that downward slope is the respiration of the system. So that's the rate at which oxygen is being consumed by everything, mostly usually bacteria. And then that upward slope is the rate at which the net rate of oxygen production. So you still have that respiration occurring, but you have this net ecosystem production in the daytime when you have photosynthesis. And then you can take the difference between those two slopes to calculate the gross primary production. So that's the actual gross rate at which oxygen was being produced in that day. And that links to the rate at which CO2 is basically being converted to organic matter in the lake. And so this has really important questions for oxygen dynamics and also food web support. It's like really the base of the food web within the lake. In terms of trying to get, so this is often used as an open water approach. So you don't really know exactly how much of what you're seeing is a signal from phytoplankton. And if there's a benthic signal, it's related to the mixing and how much production there is there and the location of the probe. So we wanted to take some other approaches to kind of balance out and get ideas of, say, what was happening just in the sediment. So we also took sediment cores, and Melissa did experiments with them looking at different levels of light exposure, so we could put them into models to get photosynthesis radiance curves from sediment cores in taken from Bear River Bay and Farmington Bay. Doug Ramsey led the charge on a remote sensing component, which he's going to present after me. And then we also took these together into kind of a modeling approach. So seeing also literature relationships with chlorophyll measurements, so looking at the biomass, pairing the remote sensing with our oxygen curve data to try and see also how chlorophyll and production related in Great Salt Lake, and put that into the model to look at what the what was happening in the seasons where we didn't have direct measurements from, or to estimate that. We also recognize that, so the 1976 paper really just looked at 2 stations in Gilbert Bay, but we wanted to get a little bit more breadth across the lake conditions. So we had 11 stations ultimately that included Farmington Bay. We worked offshore. There's Wayne and Gilbert Bay. We worked with USGS as well on maintaining and getting access to nice Gilbert Bay spots. Thank you to Christine Ramsey and those folks. We also had a station up in Gunnison Bay, which we accessed through the causeway. At the west end of Causeway, we accessed this coastal West Coast microbiolite station, where we wanted to see how different the production rates there on a nearshore Gilbert Bay site were. And then we went up to Bear River Bay, so looking at the shallow sites. And we had 3 Bear River Bay and 3 Farmington Bay sites to try and get a sense of the salinity gradients as well that were in those bays. In terms of the sampling approach, these 2 photos, I think, say it all. We were out there in the cold and in the hot weather. The original idea was to start deploying oxygen probes as soon as we started the project, summer 2020, which was a fun time to be out on boats together. And then we were planning to have it until the end of 2021, and I'll talk about what happened with that soon. And then the idea was that it was going to be too difficult to maintain them in certain weather conditions of accessing and uploading data through the winter. So we pulled them out. We pulled out the oxygen probes in winter and planned to have remote sensing to kind of fill that gap in terms of getting a sense of what was happening in the system. As you know, Great Salt Lake has seen higher elevation days. And actually in 2021, a lot of our sites simply became exposed. And Melissa had to hike out to retrieve some samples, retrieve some of the probes that had been underwater in spring. And by summer, we're not in the water anymore. There was also an issue with one of the Gilbert Bay sites in 2021. We had 2 Gilbert Bay sites planned for each year. And one of them ended up, I guess, drifting quite far. So we could only use data from a new site that we had introduced in 2021 So we had to use basically different sites for Gilbert Bay in 2020 to 2021. So we're limited in terms of what we could say in terms of continuity across that summer 2020 to the summer 2021 period. But given those conditions, there's still a lot of interesting data that came out of this. One of the first things I wanted to point out in terms of results is just looking at the chlorophyll A. So we had chlorophyll A differences that we expected in terms of having a lot more productivity in the shallow bays, these eutrophic bays, Bear River Bay and Farmington Bay. And there I'm using the productivity, but we have a lot more biomass of chlorophyll A. And then we had lower biomass of chlorophyll A in Gunnison Bay and Gilbert Bay. And I should note also that these are log chlorophyll A graphs. So we actually had really high chlorophyll A data that are not quite believable. And it was especially in the Bear River Bay and Farmington Bay, their values in the, seasonally go up to the thousands, in a couple cases, micrograms per liter chlorophyll A. And that was, and yeah, so for people who've worked with chlorophyll A in systems, that's far higher than what you would see in Gilbert Bay, which isn't that surprising, but it's higher than what you'd see normally in even hypereutrophic lakes. We believe that it was probably kicked up by the airboat that we were taking out on the bays. But we tried modifying the sampling approach by waiting for the waters to settle, and that didn't seem to change that. So I'm including the data just because we have that data, but there's a big sort of red flag there with some of that. In terms of the actual aerial gross primary production rates, it actually wasn't as big a difference as I expected. And also I was surprised to see that aerially Gilbert Bay came up actually as having very high primary production rates relative to Farmington and Bear River Bay. Again, I point out that these are aerial, so the volumetric rates would have been higher in Bear River Bay and Farmington Bay, but because these sites were so shallow, when you translate those to rates per meter squared, it actually kind of pulled them back. And when for the Gilbert Bay, you basically consider, when you're converting from volumetric to aerial, you consider the entire mixing depth that productivity is occurring within. And so that expanded those rates at an aerial basis. So you basically have a lot more water column producing and enacting that primary production in Gilbert Bay. That's creating those higher estimates. And you can see here the microbialite station as well, which is within Gilbert Bay, but was then very near shore, had extremely high rates too. And these are all rates right now that are still shown in grams oxygen per meter squared day. This is also a new figure that Erin shared with me just yesterday, where you can see a timeline in terms of the rates that are happening. And so this is August when we first installed the probes in 2020. And then you have the winter where we took them out. You can see there's somewhat different timelines. We took them out. And then we had, there was a very long delayed period before which we were able to access the lake in summer for the offshore. And then we have this actually shallower Gilbert Bay site in summer 2021 and then into fall. So you can see that there's at least seasonality as well as differences between the lakes. I just wanted to point out again that, as I said earlier, that we have a different site in Gilbert Bay, which was more offshore in 2020, and then a more near shore site in 2021. So this isn't necessarily this could be a difference in seasonality and or location differences. So spatial heterogeneity. In terms of the remote sensing, you can actually see this is one shot, and again, Doug's going to go into this a little bit more, but you can see how much spatial heterogeneity there is in the system. This is January, February 2020 versus July, August 2020. And you can see there's also, again, you might note that there's a lot of chlorophyll apparently in Gunnison Bay, but again, we don't really actually have a really good way of, we did not adjust the chlorophyll spectral data to Gunnison Bay's water chemistry. So that's a little bit, that could be a relic, but we did have high chlorophyll A concentrations measured in Gunnison Bay as well. But that does not necessarily mean production. So we know that there's a lot of chlorophyll there. We know that there's a lot of wintertime chlorophyll in Great Salt Lake, but there could be a relationship in terms of low productivity per gram chlorophyll given the wintertime cold temperatures. So that's something that's kind of a next step that would be interested in looking into. In terms of the overall model, so I mentioned that we tried to take these things together in the different lines of data that we had. I should mention, I didn't show it there too, but with the photosynthesis irradiance curves from the benthic primary production, those actually also showed relatively high rates of benthic production when you have light saturation in the shallow bays. We paired that with Secudesk measurements when we could, and with light attenuation measurements as much as we could as well. And a lot of the shallow bays were also quite turbid, so that kind of limits some of the benthic production that's in the models. And then moving forward, so what we have here in this figure is these are the diel oxygen curves, the mean data from each of the bays that we had. Again, keeping in mind that these are not from the full year, but these are just when we had them sort of grouping together all the data from each of those bays. And then in the columns, I showed what the actual annual production estimates were from the models. So making assumptions of chlorophyll differences per month using the remote sensing data that we had, and then pairing that with an average of literature estimates of how much phytoplankton production is typically associated with resulting from microgram chlorophyll A concentrations. And our own relationship that we measured in Gilbert Bay when we had daily chlorophyll A estimates from remote sensing paired with daily diel oxygen curve GPP estimates. So there's the overall kind of they're in the same range, but you can see there's definitely some differences. I think, so with Bear River Bay, I believe that the model is probably overestimating GPP. So again, we had those really high phytoplankton measurements, and those went into the model. Since there was kind of a smooth relationship over the course of the year, there wasn't an easy cutoff. But I think that this is overestimating how much phytoplankton production there likely was. And our data that was actually measured there is probably more realistic. In terms of Farmington Bay, I think it's almost maybe the opposite. So our data largely ended up being in pockets where you still had water. Whereas the model took into account that it was looking at 2020 and 2021 lake levels. And by 2021, the Farmington Bay essentially disappeared. So that's why it estimated production to be so minor across the entire Farmington Bay area. And that is not captured by our direct measurements in the pockets where we still had water from these deeper areas. So this is maybe more realistic. And then most of the productivity, when you're looking at the weighted means of the entire lake, Bear River Bay and Farmington Bay do not make that big a dent. So you really are looking at what was happening in Gilbert Bay and Gunnison Bay in terms of total lake productivity. Gilbert Bay, I think we may have had higher rates of primary production measured, especially in that 2021 data, because it was at a shallower site. So we might be getting that benthic signal. And we don't really have necessarily good parameters for how productive the Gilbert Bay benthic periphyton communities are, because we actually only took sediment cores from Bear River Bay and Farmington Bay and used those, and there were some differences even in those communities in terms of how productive they were at different light intensities. That said, I did a few calculations. You have to basically multiply benthic production, at light saturation by about 10 to close this gap in Gilbert Bay. But those rates are still within what you see in the literature for that, if we were to do that. On the other hand, with Gunnison Bay, we had just a few measurements in 2020 that indicated that it was less productive than Gilbert Bay. And in 2021, the data kind of went all over the place, and they got higher and higher and higher, and then it just seemed to go insane. So I've excluded that data even from here because it seemed to be unreasonably sort of chaotic and maybe an issue with the probe. But the models indicated that Gunnison Bay should be productive because it has so much such high chlorophyll concentrations, basically. But we also incorporated into that the light attenuation profiles that were carried out. But I would say that Gunnison Bay remains a bit of a mystery that needs a bit more study. Assuming a 1-to-1 molar ratio in terms of how much oxygen was being, in terms of the oxygen dynamics to the carbon dioxide, the carbon dynamics, this average of both approaches, both taking just the means of what we measured and the weighted means from so the weighted means from bays for what we measured and the weighted means from bays from the model approach, both came to about 1,300 grams carbon per meter squared year. And the paper that was out in 1976, the Stevens and Gillespie, was estimated about 145 grams carbon per meter squared year. So that could be due potentially to change over time. We can't exclude that. I think methodology differences are definitely going to be part of that difference too. So even in the earlier, there's a paper from 1981, Hammer, and that looks at saline lake primary productivity around the world. And at that time already, it lists Great Salt Lake's estimates from Stevens and Gillespie being at the low end of what they see around the world for lakes being typically productive. And they note that when you have bottle experiments for phytoplankton, they typically resulted in much lower primary production rates than if you have open water experiments and open water data. So we would already expect, just to having taken this open water approach, that we would have higher production rates. Not necessarily a whole order of magnitude higher, but that's, I think, safe to assume that that's part of the difference. I also noted, I was looking just through my old textbook, and I found that these rates of 1,300 grams carbon per meter squared year, they're at the high end of what you see for lakes around the world. And I looked even into terrestrial literature, and you don't see as much gross primary production, but net primary production in tropical rainforests per meter squared is about the same. So these are really astronomical rates. And here's a chart I found from an old limnology textbook, just looking at annual rates of primary production in the same units. And you can see that they go up, at this time they reported up to the thousands. So we're still within the range of what's in the literature, but at the very high end of it. So just the takeaways quickly here, we can't speak to the differences of whether 2020 or 2021 was more or less productive over that period, or even looking at the differences between the 70s and now, because we're using different methodologies. We don't have direct measurements of productivity through the winter. Gilbert Bay's winter gross primary production in the model was estimated based on chlorophyll. I'm guessing that that's an overestimate, but also Stevens and Gillespie indicated that springtime was the highest productivity period, and we don't have springtime Gilbert Bay data either. So I'm guessing that we're missing a low period, we're missing a high period. So there's not a clear bias, I'd say, either way there. The bigger question is maybe Gunnison Bay. There's a lot of unknowns, I would say. I mean, it seems to have a lot of biomass, but that's not necessarily going to result in high production. Even though, given all that, I did a quick calculation just this morning to see if we were to look for a very extreme conservative model. So if we assume that there's zero productivity in the winter and that there's zero productivity in Gunnison Bay, just with the rates that we have, we still get more than double the average annual gross primary production compared to what was reported in the 70s. So we're above the lake is more productive than it was previously thought to be, whichever way you look at it. And I would also point out that the second half of Melissa's work for her master's degree was looking at terrestrial CO2 emissions from the Great Salt Lake. And she was looking at some terrestrial vegetation productivity as part of that work as well. And absolutely, I think it's fair to say that this productivity, which is a source, again, the base of food web and a really important source for migratory birds and insects and all of the animals that Ed was studying as well, is not replaced by terrestrial plants when the lake desiccates. So you're not getting any kind of compensation really. And one of the interesting things that we were able to do, looking at the data for the diel oxygen curves, is you calculate the oxygen flux within the lake, with the lake in the atmosphere rather. And pretty much all bays that we measured had average fluxes that were having oxygen fluxing from the lake to the atmosphere. So that's negative on this. So loss of oxygen to the lake. So that also indicates being very highly productive system. So it's producing a surplus of oxygen that's going to the atmosphere. And again, that's kind of not what we expected, I think, going into this study, but it was interesting to see that as a result. And I don't know if I should take questions now, or if Doug, if you want to just go straight through and then we all take joint questions at the end.
your information into the chat. I think that might be appropriate now since we're at the end of our scheduled time, and then if folks want to stick around and ask questions, that would be fantastic. Dr. Wurtsbaugh has agreed to do his presentation next time. Since we are at the end of our time, I don't want to have anybody miss out on his presentation. So we're going to move that to the February meeting. And if I think we have a couple of last minute agenda items here.
Doug Ramsey was going to purchase on the remote sensing.
Okay, I didn't. Okay, that was part of Erin and Doug. Okay, my apologies. So, Dr. Ramsey, then I guess we'll move to you. And so, if everyone is comfortable sticking around for a few minutes, we'll move on to that.
Okay. I'm trying to share my screen, but my computer won't. The permissions won't let me share. So, let me figure this out. I actually might have to jump out and jump back in, which makes no sense at all. Oh, well, maybe this will work. Yeah, I'm going to have to jump out. So, Wayne, if you can talk, I have to recycle this system. So it's only taking about 2 minutes. Well, about a minute to go in and out. So I'll be right back.
I can take any quick questions if anyone has them while Doug's doing that.
A quick question, Soren.
Yeah.
What is the relationship between productivity and things like temperature and nitrogen concentration in the lake? Or did you guys look at any of those factors?
We didn't do nutrient data. I know that there was another team for the USGS that were doing nutrient dynamics, I think just in Gilbert Bay, but I'm not aware of any that was done in the shallow bays, which was, so we were looking at the productivity, but we haven't paired it to nutrient dynamics. I think Wayne has done previous work on that though, with nitrogen. Correct me if I'm wrong, Wayne. Let me.
Myself and several other people, including Doyle Stephen, back when looked at nitrogen as being the primary limiting nutrient in Gilbert Bay. In Farmington, phosphorus is the primary limiting nutrient. But we haven't paired up temporal variations and limitations.
So wondering if you use more nutrients or more heat from, when those earlier studies were done, and that's why it's showing such an increase in productivity.
Yeah, I mean, it probably is more eutrophic. I mean, we've been pumping nutrients in there for a long time. Well, our data is pretty highly variable. We had problems with songs going down and so forth. Doyle's, Stevens' data is just highly erratic from month to month. They did it monthly, except in the winter. And they would have to get the units 27 units 1 month and the next month 0.5.
Okay.
And some of that could be just due to the brine shrimp come in, they graze everything down. So a lot of high variability, so it's difficult to interpret some of these.
Sorry, can everybody see this? Can you hear me?
Yeah.
Can everybody see the screen?
Yes, you're good, Doug.
Okay, sorry about that. That's what I get for having a map with too much security in it. All right, so Wayne approached me, beginning of last summer, to see if I would help him model chlorophyll concentration in the Great Salt Lake using some in situ data that they had. And I agreed. So I spent a good part of the summer doing this. It was pretty interesting. We had 3 data sets. The data set that was collected by Wayne in 2006, Soren's data that was collected between 2020 and 2021, and data from the co-op, about 18 points that also came our way. So we had about 205 points that we could use to model from. And I decided to test 3 different remote sensing platforms, and I found 4 published spectral indices. My background is in the area that's kind of like anti-water. So I do deserts. So this was a little bit new to me, so I had to do a little bit of literature review to find these indices that other people have been using. For the data sets, I used 2 of the Modus products. Modus, I'm not sure who's familiar with Modus in this group, so just a quick rehash of it. Modus is a sensor. It flies on 2 different platforms. It's been flying since about 2000, and it provides daily coverage of the globe, especially our latitude. It covers us every day. It's multi-resolution, and it's been a workhorse for a lot of terrestrial, aquatic, and atmospheric products. So they generate from this sensor a number of different, a few dozen products. One of them is this MCD 4384 product. It's just a processing of the Modus imagery. Second one was this MOD 09GA product. And then I also tested Landsat to see which one worked out the best. And I won't go through all the machinations I used to figure out which one was the best. The one with the red circle was the best one. And it was the best one for a reason. This particular product is a daily cloud-free image of the globe every day from 2000 to the present. And they generate that cloud-free global composite by taking 8 days prior to the day and 7 days after the day and masking out all the clouds and snow and error-prone pixels, and then averaging the leftover pixels into one image. But they weigh the average to the day that you're interested in. So for any given day, the spectral value that you get is an average of those 16 days weighted to that particular day. The second product is just basically the image by itself. And by the way, both of these are calibrated across time. So technically you could model today based on data today and apply it yesterday to another day. So the Model 9 product is almost exactly similar, but these are just the daily images. We didn't use that because there were too many clouds, too many sample drop-offs because of clouds and everything else. So that one didn't work out as well. And Landsat had a similar problem that these images are at least 8 days apart from one another, typically 16 days, but there are 2 platforms, so every 8 days. So it gave both of these 2 latter products gave us much lower correlation coefficients compared to the MCD product. So that's what we used. Unfortunately, it's a 500-meter resolution image, which caused some other problems down the road. As far as the spectral indices, I tried out these 4, and it turned out that that last one, the blue minus red, reflected light divided by the green reflected light, ended up being the best by actually a couple of orders of magnitude as far as the correlation coefficient is concerned. So that's one we ended up using. So to identify some of the problems, these are the graph on the left identifies the ideal spectral curve for algae-laden water, the green, and for clear water, the blue, across these spectral bands. And these are typical of the spectral bands on Landsat and Modus. At least they're contained in both sensors. The graph on the right are pixels that I sampled on Utah Lake, the green line, showing a huge jump in the green, and then it drops off towards the infrared. And then when you get to the far or the shortwave infrared here, it's pretty much bottomed out to 0 reflectivity. The red is the Great Salt Lake. The brown is Strawberry Reservoir, and the blue is Lake Tahoe. So Tahoe is a very typical clear water signature. Strawberry is pretty good. Salt Lake, not quite as deep and clear, and of course, Utah Lake the same. So those are the kind of responses we expect. The problem is these are the spectral signatures So these curves are analogous to these curves on the right. These are the signatures for every single sample that we had for every single date. The one on the left are all the samples, and then the ones on the right are samples that we ended up with by winnowing those things out. So you can see these signatures up here, the blue and the yellow, very strange water signatures. I suspect these are probably dry ground or cloud, still some residual cloud pollution. This one's an odd one here. This one is very odd. Nothing in nature should do that. I'm not sure why it did that. So the process here was to identify what the problems were with these samples. And it turned out that some of it had to do with the water depth because of the other submerged aquatic vegetation. And some of it also had to do with the size of the pixel, because it's a 500-meter pixel. And in those shallow, very narrow streams that ended up occurring in the Farmington Bay area, we're getting a lot of land in those pixels. So once we cleared all those errant pixels out, we ended up with what you see on the right, a relatively clean response to that, which is more typical of a water type of signature. So the image on the left represents all the sample sites segregated by co-op, the data comp, which is Soren's data, and the sense, well, maybe backwards. I think data comp was Wayne's and Sensum was Soren's, and then a co-op points. These, the way they were distributed across the lake, they're all down in Gilbert, none of them up on top. And these are the points that I extracted, so I took away from the analysis because of water depth. And typically the water depth was in these narrow inlets because the lake is drying up. And so I was basically sampling a lot of open land. This one was on the causeway, so I was sampling the causeway too. And these are the samples that were not only less than 0.55 meters. I think I made a mistake there. But also the chlorophyll concentration was greater than 200 micrograms per liter. So they were abnormally high. So to give you an example, that point surrounded by that red circle is in water. It was taken from a boat, but it was collected. Can't remember what date it was, but that image corresponds to the same day that that point was collected. And there's a spectral signature here. So if this point was collected, say in 2006, then we would have retained that point because the water would have been a little bit deeper there, and we'd have more of a buffer of water before we hit dry ground. This is what we got as a result when we took those errant points away. We had 205 to start with. About 90 some odd were taken away because of those issues. And this is the correlation between the log of chlorophyll A and the index. So the index is basically just the spectral index and the result of that calculation and the actual in situ data on the ground. And this is the correlation between those two variables. To model it out, I used Google Earth Engine to process all the data because it's a lot easier to process temporal data with Google Earth Engine, and applied this formula. This formula corresponds to this line, and generated the estimated chlorophyll A concentration based on this index. So the result, the one on the left is the chlorophyll A output. Like Soren said, Gunnison Bay is, you really can't take that seriously, and probably not even Bear River Bay. And of course, the evaporators to the south, those are anomalies because there's no data there. We just don't know how to, we haven't been able to calibrate it yet. We did process these using a linear equation to take it from this estimate of chlorophyll A to primary production, which Soren showed you earlier. And of course, Gunnison is in this particular image looking pretty low as far as production is concerned. I also generated this for every day for 2020. So you can generate these images for every day since 2000. And the curves that you see here are the chlorophyll concentrations for each day between 2001, I think it's January 2001, it's kind of small, all the way to January 2022. So every single day for different locations, for different sampling sites. The stop motion video on the right is just the estimate of chlorophyll A across that given year, 2020. So you can see that it's pretty low in the summer. This is July now. It's moving into August. So let it go through August. And once it gets past September, October, it starts to heat up a little bit. The black areas are areas where it was masked out. There was no estimates. So as we get into October, November, you see it bloom from Farmington Bay into the lake. So spatially, it makes sense. The actual estimates are probably a function, the accuracy of the estimates are a function of the error of the correlation. So that's, I think, the last slide. Yeah. So I could, one thing I want to do is see if I can generate the same output with Landsat imagery, because technically Landsat imagery should be somewhat correlated with the MODIS imagery, the way they've processed his last products. You should be able to crosswalk them fairly easily, but I'm not sure how well it's going to work. The biggest problem with Landsat, you don't have it for every day, so it has to be a different time step. Questions?
Okay. Dr. Ramsey, if you wouldn't mind putting your contact information in the chat, I think if folks have follow-up questions, they can do it that way, but I don't see it's here or online. So as I had mentioned, Dr. Wurtsbaugh had agreed to move his presentation to next time since we're a bit over on our time today. Oh, excuse me. Okay. But on the agenda here, we did have other updates, meetings, anything else anybody ought to be aware of. I did want to point out that it looks like the Stegner Center Symposium will be held on March 16th and 17th this coming spring. And it looks like registration will open in January. Other than that, I don't have any other meetings outside of the Advisory Council and Salinity Committee and these scheduled meetings that we have discussed. So I will follow up on this meeting with an email of the Advisory Council meeting dates, the tech team meeting dates, and then some more information about the timeline for our hot topic grants. So please feel free to ask your colleagues if they have any research requests and send them on over so we can start deciding what to put on those grants. But does anybody else have any updates?
Do we have a meeting date in February?
We do. So, and I apologize for the confusion. The meeting dates that I had listed off at the start was in relation to my advisory council updates. So our next meeting is going to be on Valentine's Day on February 14th. So we can come and have a nice romantic meeting.
Same place, same time.
I will try to make this meeting in our either in our auditorium or somewhere that's not in the Core Center, so we don't have to walk through the snow. So to be decided on the location, but it'll be here at DNR on the DNR campus. But hopefully in the main building.
You just drive around, that's no problem.
It's good for some, maybe not for others. I've heard mixed reviews, but we'll attempt to secure a location here shortly.
So the hot topic's open now.
So we don't have any hot topics identified. We're taking nominations just for the subjects. So if there's a topic that you would like to research, you can send that information to me and we'll formalize that into some topics that we'll ask for grant proposals on. Okay. All right. Thanks, everyone.
Jim.